All of them. Yeah, let's say we find trees there.
Talking to Robert.
Yeah.
He's applying to become chairman of the public. He was very young guy. He was applying because he was 6 years chairman. My father is now, and then grandfather is very good. So I'm destined to become one. I've actually I heard about this initiative. They asked me he was very wrong in the critiques essentially. But Rosner said the same. He told me how he judged. He was essentially responsible for judging all his and he's a little bit inspired critiques. And then I say, okay, this is a good
We're going to meet there.
He was the only one who said, no, that's why a lot of
Anyway, I can come to
this. But you scientific talent is passed from generation to generation because it goes from father to son-in-law. Well, it's true. It's very rare when son-in-law. You can always find this thing.
It was my idea.
You know, same thing as a German system. I know that his father was a German. He was a chairman of research committee, newly formed university, Lubbock or something. No German will never get a job. But in this case, because he was married to his daughter, he made an exception. He was chairman of the German university. But you should visit Prague. Visit a statue. It's a
Yeah, I'm sure it's a million people tomorrow. Well, that's not what happened, right? That's true.
I have not seen a picture.
This would be bad.
Yeah, I mean, it was a lot.
Yeah.
It's very good picture.
Yeah, that was very artists, poets, and amazing legacy.
No, in New Jersey. I mean,
July 1? July 1. He will come home.
you know, the fun, if you talk with him.
He's a cardiovascular guy, so basically he will get another center. Only center to be cardiovascular. we have cardiovascular physician, Gene Schaefer has a center, has a center. I'm sure this new guy will tell us a little bit too many centers.
Okay, I think we can get started. So we have with us Dr. David Christini from, you can see where from, this beautiful place called Weill, Cornell, where he's a professor of cardiology and of physiology and biophysics, and of a few other things. I forget, computational biomedicine. And it's located on York Avenue, those of you who don't know where it is, Upper East Side of Manhattan. And he came to visit the Midwest for 2.5 days. So David received his Ph.D. from Boston University. And I can tell you that he's doing some beautiful research combining multiscale modeling and modeling with experimental work. And he's also a very good volleyball player. David.
Thank you, Norm. It's been many years since I played volleyball, actually, with 3 young kids. So, so I'm in New York City, and these trees here are supposed to suggest to you that we have a beautiful campus like you do. I have never been here before. I expected it was going to be a beautiful place, and it exceeded my expectation. And thank you for the invitation to come, because it's been a great day talking to you all. So I'm going to talk to you about my group's work. And as Jordan said, I'm trained as an engineer, biomedical engineer, Ph.D. And our group combines quantitative modeling and experiments. And we've been thinking a lot in recent years about atrial fibrillation in particular, and today I'm going to talk to you about some of our work in that area. And specifically, some of you may have seen a couple of our modeling studies, whole-atria modeling studies, and I'm not really going to talk about our past work in that area. I'm going to talk about stuff that's brand new and we haven't published yet. We hope to publish soon, so it's all almost all new stuff. And it really, more than half the talk is really kind of in our cellular level attempts to improve the human atrial model that we're using, because we started to have concerns that the human atrial model was not able to reproduce the dynamics at short diastolic intervals that you see during fibrillation. And if your cellular level model is not solid, then your multiscale whole organ modeling, of course, falls apart. So I'm going to talk to you about our work in that direction. I think I could skip this slide. In fact, I had removed it, but I put it back in at the last minute because Igor and I were talking earlier in his office, and he showed me these figures, and I thought it'd be okay. But I think everybody here is familiar with the differences between sinus rhythm and atrial fibrillation. And so I'm just, so we don't, just gonna buzz through and move on, and why we're interested in this problem, as many of you are as well, is atrial fibrillation is a problem that's just increasing in incidence as a function of age, and it's just increasing because of our aging demographic. It's going to be a bigger and bigger problem. 5% of the age of the population over the age of 65 has atrial fibrillation, and it has significant problems associated with it. However, the therapy approaches, the therapy for atrial fibrillation still are lagging. Pharmacological therapies are not very effective. Ablation therapy, I'm going to talk about towards the end, seemed to be better than it really is turning out to be. Cardioversion is tough. I know Igor's group is working at novel types of cardiac stimulation termination, which is very exciting. I'm not going to be talking about. So this slide is trying to justify why a multi-scale computational approach is appropriate for this particular problem. And first of all, it's very difficult to study clinically. And this particular figure, I'm not going to go through in detail, really emphasizes for non-cardiac groups, especially what the problem is here. And this figure's from one of Sanjeev Narayan's papers, and you'll see similar stuff towards the end of my talk. But basically, they record electrophysiological data from sparsely placed electrodes, and they get these electrogram mappings from these multiple electrodes. And to me, it just looks really, really difficult to make any sense of. In their algorithms, they're able to, these red lines are meant to represent propagation of a wavefront from this electrode down to this electrode, and then they interpolate the data. These are kind of splayed out atria, and they interpolate the data such that they're able to suggest the existence of a dominant rotor. But the point of this is really just emphasize how sparse the data are when using these particular types of methods. I know that Yoram's group, for example, is doing a lot of work with the ECGI that's able to provide very exciting data on atrial fibrillation. But in general, it's tough clinically. It's also tough in terms of animal models. This paper from Jean Nerbonne demonstrates why small animal models can be fundamentally problematic to use to study some certain cardiac arrhythmias, because there's a fundamental difference in the action potential morphology. We talked about that some when we were chatting a little while ago. Large animals have many of the similar problems as clinical studies, and additional ones as well, which is one of the reasons why computational model can try to kind of bridge the gap between some of these studies. It's not a cure-all in terms of the scientific approach, but it can be very useful. We in our group study it at multiple levels, and today I'm going to be focusing mostly on the cellular level and the organ level. And the cellular level, I think probably pretty much everybody in this group is familiar with what we mean when we talk about a computational model at the cellular level, but as you probably know also, when you talk to other groups, people sometimes don't know what that means. So this is a model not of an atrial myocyte. I'm going to be talking for a while in this talk about ventricular cells, because we're using those in our laboratories to develop a new approach to making a model. And this is just meant to represent, this is a schematic of a cardiac ventricular myocyte with a typical set of components that many of which you've seen. This is a series of ordinary differential equations which describe the kinetics of the sodium channel. And so I want to revisit with you the traditional model development paradigm. We're only going to be talking about Hodgkin-Huxley type formulations, not about Markov models, which some of you use. But the traditional paradigm works something like this. So you basically take an expression system like a oocyte, and you deliver a voltage clamp protocol, and from that voltage clamp protocol you get a series of ionic current recordings. And from those current recordings, you can then fit a series, and from those and a series of other voltage clamp protocols applied to the same type of cell, you can get you get these series of ordinary differential equations for that particular channel type. And then what you need to do is you need to derive equations for all of the other channel types in the cell. There's a number of problems, one of which is that no one group does every one of these experiments, and so these models are based on composites of data taken from many different laboratories under many different experimental conditions, often in different species, and then put together. And then what we as modelers in the community have often done is what we want to do is we wanted to reproduce an action potential. But to get it to do that, for example, not necessarily just an action potential, but that would be the kind of standard thing to try to fit it to. to get it to do that often requires the modeler to go back and make some tuning to some of the parameters. And we hope that that's done in a physiologically realistic way. However, the hand tuning is difficult and not always likely to adhere to the physiology. And I'm going to also try to convince you that tuning to fairly simplistic, what I'm going to tell you in my opinion, a fairly simplistic dynamics such as an action potential or a series of action potentials is what limits the ability of some of these cell models to reproduce fibrillatory activity. And so now I'm going to talk to you about an approach that we're using in the lab to improve on the model. And the approach that we've developed computationally. And so the next series of slides is our computational work on this. But everything that we've done is with experiments in mind. So we want to develop a protocol that we could do in our laboratory to apply to an intact monocyte, in this case, in the current situation, guinea pig ventricular monocytes. And we're using something called a genetic algorithm, which I think some of you have heard of because I know John and I were talking earlier, and I guess he gave a seminar a month or so ago, and you probably heard him mention that, but we're using it in a different way. And so this slide is going to step through what we're doing. And so you saw this schematic a bit ago, and what's represented up here is 10 parameters, and the parameters that we're tuning are the maximal conductances of 10 of the ion channels, because as we know, there's certain variability from cell to cell, and it's often thought to be an expression level of ion channels, which is represented in these models as a maximal conductance. And so these parameters 1 through 10 are just numbered. For the time being, we'll just say arbitrary 10 parameters in this model. We're not going to tune every parameter, we're going to just tune some of them. And what this is 0% means that we're using this is the nominal out of the literature model. And although this is a schematic from our group, it's not a guinea pig model. So the model that we're using is from Yoram's group from 2009 by first author Lifshitz. And what the model is meant to represent is our cell in the dish. So we have this model, and what we want to do is it's representing the cell in the dish, and we want to then infer exactly what the parameters of that particular cell are. And what we start out by doing is we take the model and we simulate an action potential at first. And then what the genetic algorithm does is it then says, okay, we have this is our target we're trying to fit. Now, a genetic algorithm, what it does is it takes random variations. And so in this case here, it's basically, I think, 60 individuals. These are the individuals, and what that means is that you take this model and you randomly perturb these 10 parameters, and that's an individual. And this is generation. It's I think it's 40 individuals by 60 generations. We're now using 500 individuals by 100. But nevertheless, what this shows and this heat map shows an error. And so, can you see this? So what this is are the parameters of one of the individuals at the outset, and each one of them is varied somewhat off of the 0%. So the first parameter is decreased by nearly 100%, and the second is increased, etc. And when you simulate that model, you get this blue dashed non-action potential. And what it's trying to do is reproduce this red one. The next individual has these variations, and when you simulate that, it actually does pretty well. You get this blue dashed action potential, which is near the red one. The next individual looks like this, and then there's one like this. And so this one's okay, this one's not quite as good maybe, this one's not very good, and this one's horrible. If you do these, yeah.
Can I ask a question just to make sure I understand what you're doing here? You're randomly varying these permutations of the different maximum conductances.
So it's just to establish an initial population to work with. Okay. And so we take random values for them, and then if you have a random value with these parameter values, and you were to simulate it, you would get this blue trace, voltage versus time, which is not good. What we want to do is we want to get the red trace. And what the genetic algorithm does is it takes, so I'm showing 4 here, but initially we start out with 40 or 500.
So this particular, with this model, for example, how many parameters did you really perturb? 10?
10, yeah, just
Which
10.
10?
I'll show you on most of the main conductors like GNA, GKS, GKR.
Kept yourself to
Just conductors.
membrane ion conductors.
Yeah, we're not doing anything else.
Else and not extracellular calcium cycling,
Not yet.
bounce exchanger, just the conductances of main ion channels.
Yes.
But even when you change one channel, for example,
Yeah.
you allow others to change accordingly when you fit, right? So, I mean, freely change for when you fit.
Well, initially, so far we haven't talked about the fitting yet, but yes,
Oh.
so far we're just doing random assignments, so we haven't done any
It's just
actual
assignment.
of the fitting yet.
So you change one at a time.
We changed all of them initially,
All of them.
And we just, each one is assigned a random probability, a distribution about the nominal value. So you get you get a whole population of bad models, some of which are a little bit less bad. And then what a genetic algorithm does is it borrows ideas from evolution. And what it does is it throws out the worst, keeps the best, and then sort of mates the best, and kind of has, there's all sorts of different ways you can do this, and the point is of our work is not to develop the best genetic algorithm, it's just to use this as a tool. But it keeps the best, it mates them, and then there's some random perturbations. It's basically a search algorithm to search a parameter space. And what happens is, as in this second generation here, this is showing only one individual, and it's this one's pretty good. The error function is the mean squared error of voltage as a function of time. So you take the difference between the red and the blue for each point, and the larger the difference visually, the larger the error is. And a red means a big error, and blue means a small error. And then this is the best individual error from a best individual as a function of each generation. So this is the most this slide takes the longest, the entire talk. They're not all going to take this long. But if you get this, then you probably understand most of what I'm going to talk about. So what happens is if you go through this process, and each generation you gradually try to improve, what you end up with in the last generation is most of the individuals actually, the action potential reproduces the target action potential. But there's a problem. Okay, so this shows that individual right there. And so the action potential is almost perfectly matches that we tried to fit. But the actual values are not the actual value of the cell in the dish or the model we're trying to find. They actually differ quite a bit. And so what this is what this represents is something is that the models are what's called underdetermined, and so that various parameters can compensate for one another. And you can see that pretty well here in this paper from Eric Sobey's group at Mount Sinai. And boy, this is really rendered poorly, but sorry about that. But it's basically the same idea, where these are parameter values, and this is another instance with different parameter values. Both of them make the same action potential, but if you were to look at the intracellular calcium transient, you can't really see it so well there, but there's a pretty significant difference between these two particular models. So what's happening here is that our cell, if we're to talk, I can't show you, I can give you a hand-wavy explanation, but not in 10-dimensional space, but I could do 3-dimensional space. So let's say we have 3 parameters, and the actual point in parameter space is here. What this tells us is that there are many models, maybe not a spherical parameter space, but some parameter space, many models that can reproduce a single action potential just as well as the actual model. And therefore, it's impossible to find the actual model from these data. However, this starts to point you in us in the proper direction. If we had action potential data and, let's say, intracellular calcium data, and we knew that the intracellular calcium data was the red trace here, then the algorithm would throw out the blue model and keep the red model. And so what this suggests is that we need to have more data to tune our model. And we've tried to develop protocols to do that so that we can basically shrink the space from here down. I don't think we can ever get to that point necessarily, but we want to try to get really close to it, so that our our fit model reproduces the parameters in our cell.
But there's another way to think about it, which is not all of this, but there is quite large physiological variability between action potentials and between cells. I mean, you can take two cells from the same region in the heart, and they'll give you action potentials that are not the same. They have, very different morphology and so on. So the question is whether these parameter space or this parameter volume, provides you with greater error than the actual physiological differences between cells. And if it doesn't, maybe that's okay.
So there's a robustness that's built into the design.
In the physiological system per se, there is not just a single action potential that has to look exactly like your target.
No, that's right. And so what we think is that like as Yoram said, there is going to be cell-to-cell variability. To the extent that it matters, what that suggests is that there's slightly different models for each cell. And what we can do, what we can do is
the parameter space is within the physiological range, then you probably
yeah, but perhaps building a model based on aggregate data from a bunch of cells may not be the best way to do it. It may be better to build individual models and then see and look at it another way.
I'm just saying, maybe we are asking too much. If you want to make the model exactly like the target cells, maybe you have more flexibility.
Yeah.
Than that.
So we may be asking too much, but I think we can get close. And so one of the first things that we started to think about, as I mentioned at the outset, was that we would like to be able, this is not atrial data, but this is an MAP recording showing some fibrillation, and it starts to point to what we might be able to do to these cells to try to have them explore a greater dynamic space. And what that is to expose them to different diastolic intervals. And most of you or all of you are familiar with the action potential duration restitution, which plots the action potential duration as a function of the preceding diastolic interval, and which is basically what I was just alluding to. When you have a shorter diastolic interval, you get a shorter action potential duration, and that's due to different, employing or visiting different kinetic states for various ion channels. And if we only train it by a single action potential, there's no way that we can expect that it's going to be able to reproduce this well. So I'm going to kind of cut to the chase and tell you about the protocols that we're currently using, and we're using two back to back. And the first is what I was just mentioning, which is that what we're doing is we're pacing a cell, a patch clamp myocyte, but so far I'm still just showing you simulation data. We're pacing it at aperiodically timed diastolic intervals. And we thought about, first of all, doing a fairly lengthy restitution protocol, but that takes a long time. And you want to do it as efficiently as possible, both for the health of the cell and for computational reasons. This genetic algorithm can take is pretty computationally intense. And so instead, what we've designed is we wanted to efficiently sample these various diastolic intervals, and so we came up with a stochastically timed protocol. And then What we've added onto that then is a voltage clamp protocol. So you switch from current clamp mode to voltage clamp mode. And what this voltage clamp protocol looks like a sort of arbitrary, but what it's designed to do is each phase here is designed to emphasize a particular ion channel. So we're not doing any pharmacology or any ion substitution. We have a whole cell. And so what we're limiting ourselves to is changing our voltage clamp profile. I'm going to talk a little bit more about this on the next slide. Right here. What this shows is just two portions of this. One is the first portion where it clamps as a clamp to minus 120 millivolts, and this shows the percent contribution of 11 ion channels, and number 6 is IK1. And so 90% of the current passing through the membrane at this particular clamp voltage is IK1. And so what we're going to use as our target now is a dual target. One is going to be this action potential profile, which is conceptually the same as a single action potential but longer. But the other is this output of the whole cell current. So for a model, one of our model variants to reproduce this particular current well, the only way it's going to do that is if it has a good representation of GK1. Otherwise, it's going to get this one wrong. That means it's going to throw out models. It should throw out models that estimate that parameter poorly. Similarly, this particular step here, 80% of the current is IKS. And so for the model to do that well, it's going to have a proper representation of GKS. Unfortunately, each of these was designed to try to emphasize some currents better than others. And I'm not showing everything here, but many of the parameters, not surprising to the biophysicists in the audience, just delivering these types of protocols without any other tricks doesn't enable you to isolate most of the parameters, most of the currents. But we are able to isolate a few. And so what that leads us to is this modeling result. And so, as I mentioned, this is a work in progress, so we've changed nomenclature a couple of times. And what now, instead of 0% and using percentages, we're using scaling. So 1 is the nominal model value, and these are values greater than that and values less than that. And so think of the same genetic algorithm paradigm I mentioned before, but now instead of feeding a single action potential, we're feeding 5 seconds of stochastically timed current clamp data and 6 seconds of voltage clamp data as our target, and then we're using that to try to narrow down and find a model. What this shows is the results for single action potential, the black first, and it's showing what we do is we run the genetic algorithm 10 times using 10 different random variations of initial values, and it shows the mean and standard error of the best solution for the 10 runs. And what it shows is that for some parameters, a single action potential, like finding the sodium conductance, does very well. But for others, say GKS and GKR, it's not able to find them well at all. And others, there's a wide range. However, when we switch and we now, instead of training with a single action potential, we give it this entire range, it actually nails down many of the parameters really well, some of them not quite as well, some perhaps not particularly surprisingly. But so this algorithm is able to refine the nominal model very well. So at least theoretically, it's able to work in a situation where the model structure perfectly matches what it's trying to find.
I'm sorry I'm interrupting.
No, it's quite all right. Yeah.
This is done. The experiments are done for a guinea pig.
This is not yet showing experiments. These are.
This is only the model version.
I'm about to show you the experiments. Yeah.
And in the model, what did you do? I don't remember, but the model has a chem kinase pathway and the beta.
Right. So those. So we didn't touch many.
Because they're essential because they change completely, for example, the relationship between IKR and IKS.
Mhm.
So that might be part of this
Mhm.
disagreement, I would assume, right? Because, or you didn't.
It could be.
Yeah.
So some of the, some parameters we don't touch because we have no reason to believe that we can improve on what's already there, right? But we do have reason to believe that we can, for a given cell, some of these parameters can be improved for that particular cell.
No,
But I
the
think
reason I'm asking is because it jumps at me that the IKR and IKS seem to be sort of reversed,
yeah,
and they are reversed by beta-adrenergic tone and also by some chemical effects. So they could be switched like that.
yeah.
Okay.
One more theory slide, and then I'll move on to the experiments. And this demonstrates, just if we could look at the bottom here. So okay, we have a model that fits better, but it also needs to predict better. And what this shows here, it's a little hard to see because of the colors, but this shows that this is just an inset of this graph here. So what we did was we trained it on a certain data set, but you need to be able to predict data that it wasn't trained on. And this is predicting novel data it didn't see before. And this morphology here is the nominal model, and the model that was trained on the dynamic protocol reproduces that really well, and the model trained on a single action potential doesn't do as well at these sort of irregularly timed action potentials. Okay, so now let's talk about the experiments. And so you guys, I think, are pretty familiar with this type of preparation, but we're looking at guinea pig left ventricular base myocytes, and so they're enzymatically digested, and then we come back, and I'll show you. So instead of now of fitting to a model, we're fitting to real data from a real guinea pig myocyte, and so. This is just showing the single action potential protocol, but the idea is the same because as I said at the outset, we tried to design a protocol that we could deliver exactly to the experiments. And so these are results from 3 cells, 3 guinea pig cells using the dynamic training protocol that I talked about. And I'm going to try to convince you that this looks quite promising, but you might look at it first and say, wait a second, what's going on here? And so this is, again, parameter scaling. Now, one is the nominal model conductors, okay? So we don't expect, for reasons we talked about with cell-to-cell variability or difference difference in our protocol compared to the various protocols that went into generating this model. We don't expect that necessarily this is what is in the cell. But so however, what we found promising was that in these 3 cells, for example, GK1 in all 3 of the cells and GKR in all 3 of the cells are larger than the nominal model showed. GKS is much smaller than was shown in the nominal model. And you could go through and talk about similar observations in the various parameters. But the idea here is that these cells, they're from 3 different guinea pigs from the same region, and it's suggesting that there are systematic differences between these cells and the model that's and the model. And the fact that they're moving together is not proof that they're properly being identified. But what we think is that based on the theory that demonstrates that the method can work. And these results so far are promising, and I'll talk to you about what we can do to next steps as a little bit further on, and what we can do to enhance our level of confidence. But here's another thing that demonstrates that. So this is a similar output of trying to predict novel data that the model hasn't seen before, and the novel data from the cell is here. And here, and the nominal model doesn't do so well predicting it, but the GA fit model to the dynamic protocol is able to reproduce it really well from different ones in the same cell. So we think that it's moving in the right direction, and I'm going to move on to the organ level stuff in a second. But first, I'll be talking about what we're going to do to improve this. First is that we think that we can create better dynamic voltage clamp protocols. So one thing we could certainly do is we could start to do some ionic substitution or drug blocks. So you could have a particular cell, and if you make your choices correctly, you could do a drug block, for example, towards the end of your experiment to isolate another channel well. Also, we don't tune the model's kinetic parameters at all, and that's something else that we can look into. Then what we want to do to validate is something like Eve Marder's group has done. So they did a study where they correlated mRNA level with conductances for 3 different channels, and they found a very strong correlation. So they did mRNA, and then they did electrophysiology at the channel level. And so what we could do is we do experiment on our cell, and then we can do either single cell qT-PCR, or perhaps we can do population-based protein level analysis as well to see if our results that we're finding where parameters are moving in a particular direction are consistent with what the experimental data really suggests are in the cell. And of course, the whole point of this, or one of the points of this, is to try to look at human atrial cell models. So we're working now aggressively in the laboratory to study atrial cells, human atrial cells, and we're getting cells from the cardiovascular surgeons when they're putting somebody on bypass and they have to cannulate the right atrium. They have to cut a little piece of tissue out and insert their catheter or their cannula. We get those cells and we enzymatically digest it. We're having a number of experimental problems. It's turned out to be very difficult to try to, we're not quite able to reproduce what we're seeing in the literature with resting membrane potential. So we're having a hard time with that, but we're basically getting, if anybody has any ideas, I'd be happy to talk to you about it. But basically, we're basically getting resting membrane potential something around -30, -40. We're not getting -70, and which is what we'd like to see to be able to do this. So that's something we're actively working on. But conceptually, the idea is going to be the same as everything we talked about here. That's why we're working on a much easier model to develop the approach, and then we're going to translate it to human atrial cells for our AFib work. So let me talk a little bit now about organ level stuff and what we're trying to do. I think much of this is familiar to you, but for more than a decade, a dominant paradigm for the initiation of atrial fibrillation has been that it initiates in the outside the pulmonary veins. It then you get these aberrant waves that propagate along the pulmonary veins into the left atrium, and then they interact with and create atrial fibrillation via wave block. That's something that we studied a few years ago in computational modeling with a structure that was from a human MRI, and this particular study would take a long time to go into detail on, but basically what we were interested in was the interaction with waves starting in the sinoatrial node and waves initiating the pulmonary veins. And so let me see if I'll play this again so you can kind of see it. But basically, this is 4 different projections of the left and the right atrium from 4 different views of the same simulation. And the colors are membrane potential. So red is peak of the action potential, blue is resting membrane potential. And initially what the simulation showed was 2 action potentials starting in the sinoatrial node region propagating across the atria. And then we basically artificially introduced pulmonary vein ectopic beats and then mapped the initiation patterns and what was required in terms of conduction velocity and how the atrial fibrillation initiated in various regions of the ventricle, in various regions of the atria. Let's see. And so that's a normal sinus, sinus wave propagation, and now it's relaxing. And then there's going to be another one, and then the pulmonary. You can see the pulmonary vein initiation from is going to be happening from down in here, and then eventually you'll see multiple wavelengths of kind of a traditional understanding or of atrial fibrillation is. If you look now at any, you'll see wave fronts in more than one view, and so there are multiple waves going on at the same time. And so based on this, not based on our work, but based on the understanding of pulmonary vein initiation of atrial fibrillation, this dominant paradigm of ablation developed and is widely used, where basically electrically isolating the pulmonary veins from the atria. The idea being that we're going to leave the aberrant waves do what they're doing, but they're not going to be allowed. They're not going to break through to the atrium. However, initially there was a very high efficacy reported, something like 80%, maybe higher. And now it seems to be more like people are reporting efficacy, as people have watched longer, something like 40%. So a question many people are asking is, are there alternatives to pulmonary vein ablation in terms of ablation strategies? And one strategy that we're going to talk about that we're starting to investigate in collaboration with Sanjeev and Ryan is based not around the multiple wavelet hypothesis of atrial fibrillation that I showed you quickly in these simulations, but around a hypothesis that you could sort of see in this simulation where you have a single dominant wave, a single dominant rotor in the atrium. And so I'll skip that. And so you saw something like this in my second slide. Sanjeev and Ryan at UCSD has developed a method that that locates a single dominant rotor in the atrium, and basically based on that dominant rotor, he ablates in the core, and he calls this, I don't remember what the acronym stands for, but firm ablation. He finds efficacy of around 80%, and in his hands, pulmonary vein efficacy is 45%. Now, of course, these haven't been tracked out, very, very long, so we really don't know for sure. But we became intrigued by this largely because it doesn't make sense as to why this would work, and I think Sanjeev also doesn't fully understand why it works, but it does seem to work. And the reason it doesn't make sense to us can be traced back over 100 years to this work from Mines. And what he did was he took a tortoise heart and he cut a hole in the middle, and then he initiated reentry by stimulation. And he found, as he describes here, that the reentry anchored around this anatomical obstacle or hole, and it went indefinitely. And of course, that's what we now see in all types of cardiac arrhythmias. In fact, from this was a biopsy case where basically, if you form, the idea here is that if you have anatomical boundaries in your heart, that's usually often not a good thing in terms of formation of arrhythmias. So why is it that this firm ablation seems to work if what he's doing is putting an anatomical boundary in the core? Why doesn't it instead anchor this reentry instead of eliminating it? So we're trying to understand that, and we're doing that by putting together a number of the things that I mentioned so far in this talk. What we're doing is we're getting data from Sanjeev, and first of all, the data we're using so far is we're getting restitution data recorded from patients. And so let me talk to you about a slight variation we're using in our model tuning algorithm to date. The problem is that if we use a complex ionic model such as the complex human atrial ionic models, you could run into some real computational limitations in terms of how long you can simulate that, and how well you can tune it to various data. So what we've started out with is, we started with a non-ionic model that some of you may be familiar with, this type of model where instead of having specific ion channel representations, they lump them together. It's called this Fenton-Karman model. And then what we do is we make it atrial-like by tuning it to this human atrial model from the Bears group. And so we take this model, we put it through a genetic algorithm, stochastically based genetic algorithm training protocol, not just a single action potential. And we get an atrial version of this 4-variable model. And then we take that and we want to fit that to patient-specific restitution data, the amount of action potential durations recorded from his one of his patients. And then we have this kind of atrialized model that reproduces the restitution data from a specific patient. And this shows from 3 different patients the ability to do that. And let's start at the bottom first. And what the solid black trace is a fit to his, just a numeric fit to the restitution data from 3 different patients. The red is the model restitution data before it's actually fit to that data, and these various dots are fits to that restitution curve. And you can see that it's able to fit the restitution curve very well. And at the top, we look at just at the morphology of a single action potential, and it maintains its atrial form and fits the restitution data. So we have, we think, developed an approach where we can take some real electrophysiological data from the clinic, make a patient-specific cellular model, and then what we want to do is we want to overlay that on the CT scan data structure from that particular patient, which he has. And so, for example, this is just this. Let's see here. I can find a cursor. This is just a normal activation sequence from last week for using that approach. It's a model tuned to the restitution data from a particular patient, then overlaid and simulated on the structure from that particular patient. And we hope that this is going to help us, help provide us with some insight into mechanism of the atrial fibrillation mechanism in these patients. Hopefully then perhaps some insight into why the ablation that he performed worked. And maybe long term, always a bit skeptical, as Yorm and I were talking earlier about sort of patient-specific strategies and the ability to design a strategy based on a model. But I think if we bring the clinical data and do that approach, perhaps it might help design, where exactly to ablate ahead of time. So that's what we're doing. This is that's my last slide. So what we're trying to do is we're trying to improve on a cellular level model. I talked about 2 different ways to do it: to improve on an ionic model and improve on a non-ionic model, but both using these parallelized training algorithms and complex data to make the model work better, and then incorporating that into specific structures to try to understand how the arrhythmias work. And so these are the people in my group at Cornell. And the work was really contributed by most of the people in the group. Tan Moy is a technician who works on the atrial cell the human atrial cell isolation. Tasha Lee is a very talented MD PhD student working on a different project. Margot is a post-bac programmer working on the atrial fibrillation, and Will and I did almost all of the genetic algorithm development for the guinea pig cells. Armin did some of that work. Trina is a research assistant professor that works on basically every project in the lab. Francis, some of you know, especially in that Nirvana group, is now a graduate student, but previously a programmer developing, and this, you can't really see this down here, so we developed this NIH-supported software called Real-Time Experiment Interface that about 50 laboratories around the country, including the Nirvana group and our group, and a lot of mostly neuroscience groups use to do dynamic electrophysiology protocols, somewhat similar to that which I described. And Andy Zigman helped us design our voltage clamp protocols, and Eric Sobey would collaborate on some things related to what I talked about. So I hope you understand. I relayed what we're working on, and I'd be happy to take any questions.
Questions or comments?
So I was wondering if you had a parameter when you're doing your fitting to account for the active patching. So like a lot of times when you patch it, the memory potential is higher than you'd expect than in a cell that had never been patched before.
Right.
So like kind of accounting for your probe.
So we don't, but can you systematically
Account for the
determine?
leakiness maybe?
Well, so can you systematically determine what that offset is for any given cell?
I don't know if you could, but I did notice that we always had higher resting memory potentials when we did especially the current class.
Mm-hmm. So we don't, but it's probably something we should. What we don't want to do is we never want to input just an offset current. So that's what many of these groups, some groups when they're looking at the atrial myocyte, some of the protocols actually accept the minus 40 millivolt resting memory potential, and then they deliver an offset current. To us, for what we're doing, that doesn't make sense, because if you deliver a DC offset current or an offset that's meant to shift the entire action potential, then you're perturbing things in a way that I think the model's not going to accurately represent. So we're trying to get around that, but we haven't yet cracked that. Nora?
So I have a question about the simulations that are patient-specific, let's say for the Narayan story. So his atrial fibrillation is persistent atrial fibrillation. That's where he does his work, and that's where he sees those stable rotors with the 64 basket.
Mm-hmm.
And what you're trying to do is to create an action potential that will fit his restitution, which is one property. The thing is that with long-term persistent atrial fibrillation, there's a lot of structural remodeling that is going on, and maybe that's the more important factor that determines what happens during atrial fibrillation than just the changes in the restitution of the action potential. And how are you going to take that into account in a patient-specific manner?
To the extent, so I don't think I have an answer that will satisfy you. So far, what we can do
You don't have to do this.
So far, what we can do is you take the CT scan, and so you have the structure, right? So you have some, you saw that atrium on that CT scan was pretty ugly. But what we don't have is anything at the tissue level, right? We don't have information on that. So what we can do.
Some people claim that they can see some of the structural abnormalities with MRI, but
So far, all we've done is a single a restitution from a single MAP catheter, but we haven't yet utilized any of his activation mapping. And so I think we can get some conduction velocity information, and therefore some that we could incorporate in the model. But we haven't yet gone to that. So I think that would be our next step, to try to understand some of the underlying pathology.
there's also, of course, there's electrical remodeling also.
Yeah.
So that's presumably taken into account in the restitution curve, but some of it is not, because the restitution curve is also probably not unique.
You mean it's
can
not?
have probably different combinations of ion channels that will give you the same restitution.
Yes, that's right.
So
I agree.
That's an interesting puzzle.
So I think that we could try to, we have looked at some of the electrical remodeling in one of our other studies that I pulled that figure from this figure on the right from, and that's with an ionic model. So I think that we can start with a model that more represents remodeling as well as it's understood at the time, and tune that to fit the restitution. So that would be a step closer, I think.
I think what will be interesting, what will be difficult is to get the actual structure, the substrate that has gone through structural remodeling on a per patient specific basis. To me, it seems like it's going to be a very difficult challenge.
Yeah. Igor.
The only, I agree with you that I also don't quite understand how ablating a rotor would actually help to eliminate the rotor, except for one theory which goes back to 1950s. Gurevich suggested something which might work to explain that, and specifically what he suggested was that for both atrial and ventricular fibrillation, that the rotors, or they called it differently, but in 1950s, tend to reside most of the time in areas which have the shortest, what we would now call elbow room or wavelength, but it's two-dimensional analog, basically, wavelength along the fibers or wavelength across the fibers. In other words, if you imagine the entire atria have very long wavelength, therefore re-entry will not fit there. You have a small region which, for whatever remodeling process like what Joran described, long-term physiological and structural remodeling, would have a very small, very compact wavelength. And then in this region, a rotor would fit essentially, and then the rest of the tissue will be just passively activated. So in this case, you would be able to, indeed, if you ablate this region, you would eliminate area which was capable of supporting the rotor, while the rest of the area is not capable just because of wavelength restriction. But then the question arises that in order to prove that, you really need to probe restitution properties, at least in a couple of sites, and specifically at the site of ablation. Let's say we trust what Sanjeev is doing, and let's say indeed he identified this rotor. So did he attempt to probe restitution properties at the site before he ablates versus restitution properties at some distance, which was not suspecting of supporting the rotor?
No, that's an excellent point. The restitution data I used, or we used for this, was actually from an older study of his, as opposed to one of the firm studies, just as a kind of proof of concept that you can actually do actually do all the steps. But we do. It would be very valuable to get restitution data from that site and multiple sites. And I think that he does have it from multiple sites in at least some of the patients. Yeah.
Can I ask another question from different areas? So you outline your protocol for voltage and current clamp, stochastic clamp, into isolated cells.
Yeah.
And there are pluses and minuses of doing that. And one minus which I would consider is when you isolate cells, you strip off so much from the cell on the outside that its behavior, in fact, very often doesn't look like action potentials in intact tissue. And not only because you're uncoupled from other cells, but also because you really removed all the extracellular multi-protein complexes which may modulate ion channels, pumps, etc. So I don't know, maybe it's a stupid question, but is it possible, do you think, ultimately to apply this protocol in a tissue, not an isolated cell, but tissue which is uncoupled, let's say, by specific gap junction?
I think even in tissue that is coupled, I think that you could apply parts of it. So you could apply certainly the stochastic pacing part of it, right? And so you can get your voltage data. And so the figure I showed from Eric Sobey suggests another variable that would be very useful that we should be doing in single cells, and I should add it to the to-do list, but could certainly be done in tissue level, which is calcium, right? So you could do dual imaging voltage and calcium with stochastically timed stimulation, and I think that would give you a lot of data that would at least make a model, model that should accurately reproduce the fibrillatory dynamics, if not with the exact parameters. And so to some extent, if we're interested in stimulating fibrillation, we don't necessarily have to get the exact parameters. I think this speaks back to Bjorn's earlier question. If we just want to make sure we get the dynamics right, and so that would be a way to do that. So I think that'd be a good thing to do.
I think all of these things. Yeah, it's easy to develop a model reproduce and look like very closely a single standing action potential.
Mhm.
And it's more difficult to make one that will reproduce the rate dependence of the action potential duration and the calcium transient and many other properties. And so it's number one, a question of what do you need the model for and what do you want to simulate? And number two, how many constraints you put into your model. So if and then of course there is the tissue, that's another thing. So for example, Tom O'Hara, when he developed his model, we got the data from Weill. So we tried to simulate all the whole range of physiological frequencies, both for the calcium transient, and we asked them to measure calcium transient for the whole range of physiological frequencies and the action potential duration and action potential shape at different percentages of repolarization, 10, 20, 50, 90. The more of those you put in, the more your model will be constrained. And then those measurements that were done in the tissue, multicellular tissue, we did a multicellular connected tissue strand. We're also trying to duplicate conduction losses and all of those things. So I think it's always a question of what do you want a model to do for you, and then the more and more constraint you put, the more unique the model becomes.
Yeah, so I just two comments to that. One is that I think you probably, I would imagine a frustration you have is that many people take some of your models, right, and they use them for things that they weren't developed to do, right? They use them, they push them in a totally different, we see that all the time in the literature where they were developed with a particular set of constraints, your early models with fewer constraints than your more recent models, and so people try to, but people don't, this is a model, it must be able to do everything.
They don't use it right.
They don't use it right, and so you know, your more recent models, as you were just mentioning, introduce more constraints, which makes them applicable to a larger range of problems. But at some point, a person, you know, no matter how good they are as a model, we run into the limit of how many constraints that they can actually fit. And so one of the points that we're trying to get across here is that if you use an automated tuning approach, such as that we're using and such as John is using, that limitation goes away, right? The limitation of the ability to so now you can use as much data as you can collect, as long as it's good data, and it makes the model. So I don't think you have to ask the question, you know, what constraints matter to you. I think that we should ask the question of which constraints can we eliminate by developing particular protocols and implementing them. And I think that we scratching the surface in terms of what we can do.
But there is always the other, for example, what will be the sodium accumulation after 5 minutes of pacing in a certain rate? And you can always add more of those into your mix. The other problem is that the data are obtained from, it's not the physiological system where you get the data. It's always an experiment that's done in some sort of preparation, and then naturally people are trying to use the model to predict what will happen in the physiological system. It's always an approximation, but it's probably the best we can do.
Yes.
In terms of pursuing the human cellular data, it seems like everybody is able to take atrial appendage tissue because that's what's available from surgical cases. So are the cells different in the appendage versus the free wall? And when you do simulations, do you see differences in the way it depolarizes or repolarizes? And I mean, do you ever actually see rotors within the appendage itself? I mean, granted, a normal appendage is relatively small, but you get these huge, abnormally dilated appendages in disease.
Yeah. So, you point out a limitation, which is that the cells that we have available are from one region, and it's, so therefore it falls under the same concern where it's an approximation, and we know that it must have limitations when you then because basically we were using one cellular model in different regions, at least in this particular study. So it's something that that's we're limited right now by the cells that we have available to us. But you point out a good limitation. Yeah.
So I'd just like to follow up with Jorm's question. I have my own questions or that I don't understand because I'm not doing so much of modeling. I haven't thought about this thoroughly. So we always know that we know that the cells are different. Actually, there are more and more studies to show that even the same populations of the cells, they can have different composition of ion channels to give you similar action potential, for example, or some of the integrated properties can be similar, but their individual composition of ion channels and other parts are different. So there are diversities, and people can see that they can converge to the same property. And what you are doing, actually initially I was very excited to see that you can actually, maybe you can use your genetic algorithm to actually create similar kind of a population, but then they can all, when they put together, they can converge to the properties that you try to look at a certain level. But what I felt then, what you're doing, trying to do, is that you use as many as the different properties, and then try to come up with a homogeneous model that can represent all. Is that what you try to do?
No, I wouldn't say that's our goal, to have one homogeneous model to represent all the cells. Our goal in that part of the talk was to have there is a cell, and at that point in time, when you do the experiment, it has properties, and our goal, which is unattainable, is to determine what those parameters are. And so we want to get as close as possible to those particular parameters. And I wouldn't have necessarily expected that the cells that I showed from the guinea pig would have had the movement in the same direction for these particular parameters, but they are from the same region, and so I think that. So I wouldn't have necessarily predicted that. I might have predicted that they would be quite different. And if they were quite different, I basically have to say, well, this is what it is, and we have no idea if it's right until we actually do some of the mRNA or protein level analysis. Except to say that we know that it reproduces the data better when we predict novel sequences. So our goal is not necessarily to come up with a. I would say there's different goals depending on the project, but the goal is not necessarily to come up with one model that reproduces the data from all the cells. We don't think that's going to be true. Well, what we could have, if you have enough cells and you have variability and you have all these different models, you could start to ask the question, how important is the variability? What does the variability really mean? Is it robustness, or is it a property that does it just not really matter? And so so I think that there are a number of questions that you can begin to ask as we acquire data from more cells, and once we know with a high level of confidence that the values are right.
Okay, I think we have to end this wonderful discussion, so thank you very much again.
Thanks, Ron.